A very good morning, dear students. Today we will see TCA cycle, that is tricarboxylic acid cycle. Tricarboxylic acid cycle is the next metabolic pathway after glycolysis, and this tricarboxylic acid cycle was completely described by a scientist known as Hans Krebs. Therefore, the cycle is otherwise known as Krebs cycle. And later it was identified that citric acid is the tricarboxylic acid described by scientist Krebs. Therefore, the cycle is otherwise known as citric acid cycle. So, the same pathway is known by different names. One TCA cycle, Krebs cycle, and citric acid cycle. And this is a final oxidative pathway for carbohydrates, fats, and proteins. And this pathway gives reducing equivalents like FADH2, NADH, and 1 GTP, which is equivalent to 1 ATP. And coming to the citric acid cycle, it starts from the compound acetyl-CoA. Acetyl-CoA coenzyme A. The pyruvate formed from glycolysis is converted into acetyl-CoA by the enzyme known as pyruvate dehydrogenase complex, which is the link between glycolysis and TCA cycle. Once after forming this acetyl-CoA enters into mitochondria and it become a part of this citric acid cycle. All the enzymes of this TCA cycle are present in mitochondria. Therefore, the TCA cycle occurs in mitochondria. Acetyl-CoA, a 2-carbon compound, combines with oxaloacetate, which is a 4-carbon compound, to form a tricarboxylic acid known as citrate. This is catalyzed by the enzyme citrate synthase. And here it expels coenzyme A. So in the first step of TCA cycle, acetyl-CoA combines with oxaloacetate to form citrate, and this is catalyzed by the enzyme citrate synthase. In the next step, citrate is converted into or isomerized to isocitrate through a 2-step process. In the first step, citrate loses 1 molecule of water and forms cis-aconitate. And it is catalyzed by the enzyme aconitase. And the same enzyme catalyzes for the conversion of cis-aconitate to isocitrate. Same aconitase catalyzes for the conversion of cis-aconitate to isocitrate. Here, 1 molecule of water adds to cis-aconitate. Okay, here dehydration and here rehydration. In the first step, acetyl-CoA combines with oxaloacetate to form citrate, which is catalyzed by the enzyme citrate synthase, and then citrate is converted into isocitrate by the enzyme aconitase, and this is through an intermediate cis-aconitate. Then this isocitrate is converted into alpha ketoglutarate by the enzyme isocitrate dehydrogenase. Here also on intermediate forms, which is oxalosuccinate. Isocitrate is converted into oxalosuccinate, and oxalosuccinate is converted into alpha ketoglutarate with the one carbon dioxide is eliminated. And here one high energy compound is formed. Nicotinamide adenine dinucleotide is converted into NADH, a high energy compound. And this alpha ketoglutarate is converted into succinyl coenzyme A by the enzyme alpha ketoglutarate dehydrogenase complex. This reaction is very much similar to the conversion of pyruvate to acetyl-CoA. And so alpha ketoglutarate is converted into succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase complex. This reaction is similar to the conversion of pyruvate into acetyl-CoA. And the conversion of alpha ketoglutarate to succinyl-CoA requires 5 coenzymes. That is, for the functioning of alpha ketoglutarate dehydrogenase complex, 5 coenzymes are required. They are thiamine pyrophosphate, FAD, NAD, lipoic acid and coenzyme A. The succinyl-CoA is converted into succinate by the enzyme succinate thiokinase. Succinate thiokinase. Here, one GDP is converted into GTP. Here the ATP production is in the substrate level, therefore it is otherwise known as substrate level phosphorylation. The succinate is converted into fumarate by the enzyme succinate dehydrogenase. Succinate dehydrogenase. Here one FAD, flavin adenine dinucleotide, is converted into a high energy molecule FADH2, which attaches into electron transport chain and produces 1.5 ATP. This fumarate is converted into malate by the enzyme fumarase, and this malate is converted into oxaloacetate by the enzyme malate dehydrogenase. Malate dehydrogenase. So this is the oxaloacetate is regenerated and again it combines with acetyl-CoA and continues the cycle. Here one carbon dioxide is eliminated in the step by the conversion of isocitrate to alpha ketoglutarate and by the conversion of alpha ketoglutarate to succinyl-CoA. So 2 carbon dioxide are eliminated. Finally, again 4 carbon oxaloacetate is formed. 2 carbon compound acetyl-CoA combines with 4 carbon oxaloacetate to form 6 carbon citrate by the enzyme citrate synthase. Then citrate is isomerized to isocitrate by the enzyme aconitase through an intermediate cis aconitase. Then isocitrate is converted into alpha ketoglutarate by an intermediate, it is oxalosuccinate by the enzyme isocitrate dehydrogenase with the production of a high energy molecule NADH. Then alpha ketoglutarate is converted into succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase complex with the production of one high energy molecule NADH by the elimination of carbon dioxide. Then succinyl-CoA is converted into succinate by the enzyme succinate thiokinase, the production of one GTP. Succinate is converted into fumarate by the enzyme succinate dehydrogenase with the production of a high energy molecule FADH2. Then fumarate is converted into malate by the enzyme fumarase. Malate is converted into oxaloacetate by the enzyme malate dehydrogenase. So coming to the energetics, one NADH is produced from this step, that is conversion of isocitrate to alpha ketoglutarate, one NADH2. Then one NADH2 is produced from the step conversion of alpha ketoglutarate to succinyl-CoA, and another one NADH2 is produced from conversion of malate into oxaloacetate. So there are 3 NADH which enters into electron transport chain and produces 2.5 ATPs, 3 NADH, therefore 3 into 2.5, that is 7.5 ATPs. 1 FADH2 enters into electron transport chain and produces 1.5 ATPs. Then 1 GTP, which is equivalent to 1 ATP, that is 1. That is total 10 ATPs are produced from a molecule of acetyl-CoA. From a single molecule of acetyl-CoA, 10 ATPs are produced. From glycolysis, glucose is converted into 2 molecules of pyruvate. Therefore, 2 molecules of pyruvates are converted into 2 acetyl-CoA. Therefore, through glycolysis, after glycolysis, it forms 2 acetyl-CoA. Therefore, total 20 ATPs are produced from citric acid cycle by the oxidation of glucose. So this energetics you must remember. And coming to the regulation of citric acid cycle, isocitrate dehydrogenase and alpha-keto dehydrogenase complex are allosteric enzymes which regulate citric acid cycle. Isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase complex are the 2 enzymes which regulate this citric acid cycle. The citric acid cycle is activated by GDP and it is GDP or ADP, and it is inhibited by ATP, NADH, etc. Okay, allosterically activated by GDP or ADP, then it is allosterically inhibited by ATP and NADH. So that is the regulation of citric acid cycle. Regulation as well as the energetics. You must remember this. And this is citric acid cycle. So citric acid cycle is the series of reactions which converts acetyl-CoA into 2 molecules of carbon dioxide and by producing 10 ATPs. So citric acid cycle is the series of reactions which converts acetyl-CoA into 2 molecules of carbon dioxide by a series of reactions. And totally 10 ATPs are produced from a single molecule of acetyl-CoA. The regulating enzymes are isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase complex. These are the allosteric enzymes which regulate TCA cycle. Okay, thank you.